Good morning. I'm David Miller. I'm a neurologist at the UCL Institute of Neurology and National Hospital for Neurology at Queen Square. And I've been seeing people with MS for over 30 years. It's my main clinical interest. And in terms of research, I've been involved using MRI scanning to try and better understand MS, to improve the diagnosis. But particularly and recently, and what I'm going to talk about today, is to use MRI scanning to try and bring forward, detect effective new treatments. And I want to acknowledge the support of the MS Society. It's been a major funder of the research scanner for many years at Queen Square. So, what are we trying to do when we develop a disease-modifying treatment for MS? And I think there are three broad aims. We want to prevent the relapses, if someone has a relapsing remitting form of MS. We want to prevent disability, whether it occurs from a relapse or, as is often the case, of course, it is an increasing problem during progressive MS. And finally, we want to be able to improve function. So if someone has some disability, we want to be able to sort of restore better function. So I'm going to focus on each of those three. Now, the specific title is using MRI to develop neuroprotective and neuroreparative treatments, and they apply to preventing disability and restoring function. But I'm going to talk, first of all, about preventing relapses, because you can see from that how MRI has been very useful, and then translate to where in the future I think it will be useful for preventing, finding treatments to prevent disability and restore function. So we want to prevent relapses, and one thing that is quite clear, and this has come from doing serial MRI scans over many years, is that it's a new lesion that causes a relapse. It happens to occur in a particular location, such as in the optic nerve, causing optic neuritis, or in the spinal cord, causing some weakness or numbness in the arms and legs. But there are also, it's clear if one does monthly scans with MRI, that there are about 10 new lesions for every single relapse, and that's because a lot of new lesions occur in a silent part of the brain. So this is then clear that one is seeing a lot going on that isn't apparent in terms of symptoms. And this is an example of monthly scans using an injection called gadolinium, which shows the active, the new inflammatory lesion, and they're the white spots we can see on those scans. And you can see that on each monthly scan, there are about 5 or 6 or 7 new lesions occurring. And over the course of the 6 months, this person only had a single relapse. So that has implications for doing a trial of trying to find a treatment to prevent relapses. And if of course, the ultimate outcome measure is how many relapses does a person have. But if you want to do a trial to prove that a drug prevents relapses, you need to look, it's a very big, expensive business. You need to have some people taking the drug, some taking a placebo, because you have to compare it with the standard relapse rate. And then it needs about 600 people over 2 years to be studied. So it's several million pounds. On the other hand, if you just look at trying to prevent the number of new MRI lesions, you can do that in about 150 people over 6 months. So that is the way new drugs are screened for relapsing remitting MS. And this process started 10 or 15 years ago. And here's an example of how it worked in practice. This is a drug that is, some of you will have heard of, some may even be taking it, called Natalizumab, or Tysabri, its trade name. And in the early trials, MRI trials, as this slide shows, over 6 months, it was profoundly effective. It reduced the number of new lesions by 90%. But that trial compared to placebo, so people on placebo had an average of 10 new lesions after 6 months. People on Tysabri had an average of 1 new lesion after 6 months. That trial was done 10 years ago, but it gave the clue. If a treatment's stopping new lesions, it should stop relapses, but the trial wasn't big enough to prove that. So it still has to go on and do the bigger trial, but at least you have confidence that you've got a treatment that will be proven to be effective, rather than perhaps testing 5 drugs where you don't have any proof from MRI and find they're all negative, so there's been a lot of cost and time and waste. So that's the pipeline now for relapsing-remitting MS. A quick trial with MRI to show that a treatment can prevent new lesions, the longer trial to show that it prevents relapses, and then a couple of years later, hopefully, the drug comes to market. And there are now 5 licensed treatments for disease-modifying, for relapsing-remitting MS, and we expect another 4 to come available in the next 2 to 3 years following this pipeline. But now I'm going to switch to the neuroprotection and repair. And so, what about trying to prevent disability? Well, disability is due to the loss of nerve cells and nerve fibres. That's what causes an irreversible disability. And so we need neuroprotective treatments to stop the loss of those nerve cells and fibres. And there are now a number of neuroprotection trials underway here at UCL Partners. The slide at the bottom just shows an example. The little black dots, that's a spinal cord tissue slide, and the little black dots are nerve fibres. And you can see they have a smaller density in this person with MS because of the loss of nerve fibres. That's what we want to prevent. We want to prevent that happening, start with the situation on the left and keep it like that. Well, to show that you can stop someone getting more disabled is an even bigger trial than in relapsing-remitting MS, because disability is so variable, and for some people there's no change over many years, and the rate of change is highly variable. The measurements, the clinical measurements, are very rough and ready. And so really, one is looking at a £10 million trial, sometimes more, over 3 years of about 800,000 people with MS, some on placebo, some on the drug. So major commitment. And as an example of a recent trial that did this was oral cannabis here in the UK, in progressive MS, done with very appropriately. There was some experimental evidence from small experimental models, but there was no MRI data, so they went straight ahead to a big trial. Sadly, it was negative. So if one had some way of screening, again, as in relapsing-remitting MS, it might be a more cost-effective way of developing treatments to prevent disability. So loss of nerve tissue can be measured on MRI, and one can do this in using a measurement of what we call brain atrophy, loss of tissue, and one can do a trial for about 1/10 of the cost of a clinical endpoint, a disability endpoint measure, and can study around 150 patients over 2 years. And this is what's being measured. So this is a little more demanding in terms of machine precision and image analysis than counting new lesions in relapsing-remitting MS. It does require very good precision of the scanner, but what you can then take is 2 scans, say a year apart, register them, and show exactly how much tissue is lost. And the red and blue areas are at the outside edge of the brain, and also in the inside. In the inside of the brain, one has the ventricles, which contain fluid, and you can see how the loss of tissue is occurring both on the outside and the inside. And in MS, on average, there's a loss of about 0.5 to 1% of brain tissue per year, which is about 5 times the rate of tissue loss that we all do have. So there are a number of drugs now just having started or about to start trials of neuroprotection, drugs that you'll hear more about this afternoon, where there are quite good reasons for thinking that they will be effective, but it has to be proven. And these are all going to be tested in these smaller trials here at UCL Partners using MRI to determine whether it prevents the loss of nerve tissue. My colleagues, Dr. Chataway, Dr. Kapoor, and Professor Giovannoni are leading these trials, and truly UCL Partners is leading the world on this. And I don't think there are any other drugs being tested anywhere in the world at the moment, and so here are 6 being tested in London. And the last thing I want to mention is restoring function. And I'm going to just pick up on the theme that Ahmed mentioned about trying to measure remyelination on MRI. Remyelination will improve the conduction of those nerve fibres. You saw from Maurice's talk how demyelination causes a block of conduction. Remyelination will restore function, and it will also it'll also give neuroprotection because the nerve fibre is going to be rather more immune to further attack. Now, MRI can measure remyelination, and the technique is called the magnetization transfer ratio, or MTR. And this slide shows how it works. Very simply, the MTR tells us how many tissue molecules there are. So these are the structural tissue molecules in the brain, and particularly how much myelin. Myelin is a very densely structured tissue full of proteins and lipids. And so it's very sensitive to the myelin in the white matter of the brain. And if you look in the top slides, in the normal situation, every nerve fibre has a round coating of myelin. That's the blue rings, quite dense. In demyelination, that's lost. And so you can imagine the MTR drops from, say, about 40% to 20%, something like that. And then in a remyelinated lesion, the new myelin that comes back isn't quite as good as the old. It's about half the thickness. And so the MTR comes back up to somewhere between, say, around 30%. And here's an example on the bottom of what you can actually measure. These are MTR scans of two little lesions that have been followed over a year or so, and the lesions are little black holes. But the final scan is a subtraction of the two. And if the MTR has gone up, it's green, a green-blue colour, the one on the left, that's remyelination. If it's gone down, it's red, and that's demyelination. So we can measure that now. And there are two trials underway looking at this. There's an international stem cell trial in relapsing remitting MS. And, people in that are going to have the MTR measured to see if it can achieve remyelination. And there's also another trial called an anti-LINGO antibody that's being done in optic neuritis. So to summarise, MRI is a quick and objective way to show whether new treatments can prevent neuroprotection and remyelination. And we know and we expect that treatments that cause neuroprotection will prevent disability, and that treatments that enable remyelination will restore function. So I'm sure this is the right focus now for therapeutic research in the next 5 to 2 years to target, develop these treatments, and we'll be using the scanning to try and make that whole process as efficient as possible. Thank you.